Barrows. I am the director of the UWAB Astrobiology graduate program. And I'd like to welcome everybody here to the second lecture in a series that we are holding to celebrate 25 years of our program. So astrobiology is a massively interdisciplinary science that is about the study of life in the universe, its origin, its evolution, its distribution, and future. So astrobiology helps us to understand where alien life might be beyond the Earth and the best way to go and look for it. So tonight's speaker is Dr. Rika Anderson. I'm delighted to introduce her. She is an associate professor at Carleton College and a microbial oceanographer. She will use her considerable expertise in this area to take us on a tour of the very depths of the ocean, back into deep time, and then up and out into deep space. She told me about the tour because there's a lot. It's going to be like a roller coaster ride. So Rika is an alumna from our UW Astrobiology program, so we're very proud of her. She worked with Professor John Barrows, who's also here in the audience, and she looked at the interaction between viruses and microbes in hydrothermal vents. She is, in fact, one of the first students to graduate with annual dual title degree in astrobiology. And I noted at dinner tonight that she's, in fact, now a mid-career scientist. So it's like, wow, we're all growing up. So she earned that degree in 2013. So she's no longer an early career scientist. So I remember Rika really as one of our superstar students, as she was just so incredible at understanding different sciences, gathering the bits and pieces she needed from them and synthesize them to answer completely different questions and go in new directions. And so you'll certainly see her zigging and zagging tonight. So since her time with us, she has done research at the University of Illinois Urbana-Champaign. She's been at Woods Hole Oceanographic Institute, where she actually worked with one of our very first graduates in the program, Julie Huber, who is a senior scientist there. After these postdoc experiences, she became biology faculty member at Carleton College, where she remains. She's primarily interested in that facet of astrobiology that studies how life co-evolves with its environment, how the two things interact with each other, both at deep sea vents and also throughout the past history of the Earth. And so you'll see some of both of those tonight. Rika is now also a task lead in my virtual planetary laboratory NASA research project, where she works with astronomers, geologists, biologists, biogeochemists, and atmospheric scientists to better understand how early life on Earth modified its environment in potentially detectable ways. So we'll learn about some of that as well tonight, as well as some of Rika's many other interests. So without further ado, I will cede the floor to Rika, who will take us from the deep ocean to deep space.
All right, thank you for that introduction. Thank you all for coming. It's really nice to be back in my old stomping grounds. I'm going to start by first acknowledging that this talk was originally supposed to be a joint talk with me and Jada Arni, who also came from this program. And the way we had originally pitched it was to go from the deep ocean, which is me, to deep space, which is Jada. But now Jada's not here. But I'm still talking about the deep ocean to deep space, so you'll forgive me for not being an astronomer, but I'm going to do my best. And I think I can go from the deep ocean to deep space. No problem, right? We'll just it's a small topic here, so we'll figure this out. Do I need to do anything AV-wise? There's an idea. I don't have to sound like this the whole time. While we're fixing that, I'll yell. Does anyone know what this is? Thank you. I've just divided the audience between the old people and the young people. I showed this to my students who are about 22 years old on Monday, and they looked at me blankly, which made me sad. And then Vicky called me old, which made me even sadder. But this is in fact the pale, oh boy, okay. I think you can hear me now. Okay. So this is in fact the pale blue dot image. So for those of you who are, young, this image was taken in 1990 by one of the Voyager spacecraft that was sent to explore the outer reaches of our solar system. And as it started to move to the outer reaches of our solar system, Carl Sagan, because he was Carl Sagan, asked NASA, can you please turn that spacecraft around and take a picture of our planet? And NASA said, why would you do that? It's not scientifically useful. And he said, just do it. And because it was Carl Sagan, they did. And this became one of the most iconic images of the 20th century because it was really the first time we had seen our planet in such a cosmic perspective. So I like to start with these images because it shows you how astrobiologists tend to think. We think of our planet within its cosmic perspective. And I like to show this quote by Carl Sagan himself, where he states, "If you wish to make an apple pie from scratch, you must first invent the universe. The nitrogen in our DNA, the calcium in our teeth, the iron in our blood, the carbon in our apple pies were made in the interiors of collapsing stars. We are made of star stuff." And then he goes on to say, "We are the embodiment of a cosmos grown to self-awareness. We have begun to contemplate our origins, star stuff pondering the stars." And that, I think, is what astrobiologists are doing. We think of ourselves as being born in the interiors of stars. We're trying to understand where we come from, and we're trying to understand how we fit within this universe that we live in. Are we alone? Could we find other life elsewhere? How did life even begin on this planet? So I'm going to try to take you, as promised. Oh, just by the way, here's an updated version of that pale blue dot. This was taken by, I think, the Cassini spacecraft a few years ago, looking back at our Earth from below Saturn's rings. And I'm showing this to you partly because it's a gorgeous image, but also because people are still asking these questions. We still see Earth from this perspective. We're just always updating it and thinking about how, with our new technologies, we can see ourselves within that cosmic context. But I'm an oceanographer, so we're starting here. And my title was From the Deep Sea to Deep Space. So I'm starting with where I started, which is deep sea hydrothermal vents. That's what I studied when I was a graduate student here at the University of Washington. What you're looking at here are some hydrothermal vents that we sampled at the East Pacific Rise. I'll tell you a little bit more about these vents in a moment. But let me first tell you why I'm interested in hydrothermal vents, why I came to the University of Washington in the first place. Is because by studying these vents, by studying the deep sea, we can actually get a better understanding of our own planet, our own origins, and the possibility that answer the question of whether or not we're alone in the universe, which seems like a tall order for hydrothermal vents, but I promise I'll get there eventually. So, like I said, I'm an oceanographer, and one of the things I learned when I was a graduate student here was how many parallels there are between doing studies in the deep sea and studies in outer space, because both are really inhospitable environments where humans do not do well. But we want to study the things that exist there. So we have to build all kinds of contraptions to go out there and take samples. If you're an oceanographer, we're using submersibles like this. This is the submersible Alvin that first discovered the Titanic. It first discovered deep sea hydrothermal vents. This is a human-occupied submersible that we go in down to the bottom of the ocean to take samples from things like hydrothermal vents. Similarly, we have a robotic submersible. One of them is called Jason that we use to take samples without humans inside, which is frankly more convenient because robots don't need to pee. And we send these things to the bottom of the ocean to collect these samples for us. These same conversations are being had about sending robots into space to collect samples from Mars. Do we send humans? Do we send robots? Which one is better? The things that don't have to pee are often better. But it's interesting how many parallels there are between working in the deep sea and working in outer space. So when we go to sea, we're going on these large research vessels, we're climbing inside these submersibles, we're questioning our life choices. We go down to the bottom of the ocean and we're taking these samples from these really incredible systems where life thrives. And when we get down there, we see something like this. So this is a black smoker. Well, we call them black smokers because they look like they have black smoke coming out of them. That's not smoke, that's water. It turns black like that because when seawater moves through cracks and pores and fissures in the seafloor, it comes into close contact with the cracking front of a magma chamber. It becomes superheated. Chemical reactions will enrich that water with things like carbon dioxide and hydrogen and iron and sulfur. And that superheated water will come back up to the seafloor and form these gorgeous chimneys like this. These particular vents are off the coast of Cuba. They don't exist anymore because the day after we took this video, there was a 7.7 earthquake that knocked them all over. And we sent the submersible down the next day and we're like, it's dust now. They grow back, it's fine. But it tells you a little bit about how dynamic these systems are. So I'm gonna first go through and show you a few different videos of what these vents look like, because one thing I want to impress upon you is we tend to call these extreme environments. And people think that life can't possibly persist in what we call extreme environments. They're only extreme to us, but we are measly humans. If you look at the diversity of life that live in these systems, these are oases. There's tons of life, and the diversity that lives both in a single hydrothermal vent and across the globe is really incredible. These are hydrothermal vents at the East Pacific Rise, so off the coast of Central America. You can see that these are covered in things like massive tube worms and mussels and clams and crabs, and you'll see a fish go by in a minute. I don't know, charismatic megafauna of some sort. But the thing that powers these ecosystems, the basis of these ecosystems, are the microbes. There's no plants here. There's the fish. There's no plants here. I don't actually care about the fish. There's no plants here that form the bottom of this food chain, right? It's the microbes that are consuming compounds that are coming from the earth itself, turning that into an energy source that then feeds the rest of this ecosystem. And those are the things that I'm interested in. Who are those microbes? How do they work? How do they change over time? Do they get infected by viruses? What are those viruses doing? How do they affect the animals here? Those are the kinds of questions we ask about these hydrothermal vent systems. Often those microbes are invisible, but not always. I like to show this. This is a hydrothermal vent off the coast of Sicily, I think. And I like this one because the microbes become macro. You can see the microbes that I study. Those slimy bits, those lovely slimy bits, are the microbes that live in these systems. But then again, this is another hydrothermal vent. This one's again off the coast of Cuba. And this one I think was creatively called Shrimpocalypse. There's one next to it called Lots of Shrimp. The one next to that was called Shrimp Hole. We're really creative in naming our vents. These are off the coast of Cuba, like I said. And again, you can see how different the life is in these systems. So there's this huge diversity of life that exists in these systems, and it is flourishing. It's not extreme to these shrimp at all. They love it, right? And then again, I've only been showing you hydrothermal vents that are powered by magma. But in 2005, scientists here at the University of Washington discovered an entirely different type of hydrothermal vent in the middle of the Atlantic Ocean. This one is called Lost City. It's perhaps the most spectacular example of hydrothermal vents that are powered purely by chemical reactions. So we have seawater moving past certain types of rock, creates these chemical reactions called serpentinization that creates these huge, gorgeous structures with chemistry that is completely different from the vents that I just showed you. That fluid sometimes has the same pH as toilet bowl cleaner. And you don't see nearly as much macrofauna. In fact, a colleague of mine likes to say it's the microfauna that become macro in these systems. And these are particularly interesting for studies of the origin of life. And I'll get to that in a minute. The reason I'm showing these all to you is that one of the things we can learn from hydrothermal vents is that they may exist in other worlds within our own solar system. So it's entirely possible that there are regions where life could potentially exist in other ocean worlds within our own solar system. So what I'm showing you here are some examples of other ocean worlds within our own solar system, and I'm going to focus on two of them, Europa here and Enceladus, tiny little Enceladus up here, which have some of the largest ocean worlds and may in fact have deep sea hydrothermal vents at the bottom of them. So this is Europa. It's one of the moons of Jupiter. It has a massive cold ocean covered in a crust of ice. In fact, if you were to look at the total volume of water, if you were to look at the total volume of water in Europa compared to how much we have on Earth, it would actually have more water than we do. Now it won't go forward. We're just gonna stare at water for a while. The clicker should work. Okay. Magic. Okay, cool. So if you were to look at a cutaway of Europa's ocean, it might look something like this, where you have this thick icy crust along the top, that hard Europa icy shell that you see up here. You would see a really deep salty ocean, and it's very possible that we may see hydrothermal vents at the bottom of Europa's ocean. And it's not just Europa. One of my favorite moons is Enceladus, which is one of the moons of Saturn. And it similarly has an icy crust with a salty ocean and also may have deep sea hydrothermal vents at the bottom. But this one is particularly interesting because Enceladus is spewing its guts out into the solar system. And we think, and by we, I mean people who are not me, astronomers who study these kinds of things, planetary geologists, think there may be something akin to hydrothermal vents at the bottom of Europa's ocean, powering these essentially massive geysers that come through fissures in that icy crust and emerge at the surface of Enceladus to be released out into the solar system. In fact, Enceladus forms part of Saturn's E-ring and contributes to one of its rings. And this provides this fantastic opportunity to send satellites through this spewing geyser mess, sample it, see what kinds of chemicals are there, and try to infer whether that's the right mix of things you'd expect to see from a hydrothermal vent at the bottom of Enceladus's ocean. So far, signs point to yes. This says nothing about whether there's actually life in those vents, if they do exist at the bottom of Europa or Enceladus, but it's intriguing, and it's worth sending out satellites to learn more about what might lie below the oceans of both these ocean moons. But I'm actually going to bring us back to Earth for a minute and think about what we can learn by studying hydrothermal vents on our own planet, because hydrothermal vents not only may be potential sites for habitability elsewhere in our solar system, but may also have been sites for the very beginnings of life on our own planet. And actually, in order to understand that, we have to think about what our planet looked like 4.5 billion years ago during what we call the Hadean Eon. I think this context is really important. This is not a photograph. This is an artistic interpretation. One thing to know is that the Earth probably would have spun faster on its axis. So days would have been something like 12 hours long. So if you can imagine 6-hour days, 6-hour nights, just like spinning, you think your days are crazy enough. Imagine living on the Hadean Earth. The Earth was probably mostly ocean, although depending on who you talk to, there's huge controversy about this. But largely, I think the current thinking is that the continental mass, if it existed, was quite small at this period in time. The sun was fainter. I have learned from my astronomy friends here that apparently, as stars age, they get brighter and brighter, which means that our own star, when it was younger, was much fainter. What does that mean for our planet? It's kind of an open question. We're losing our moon, sorry to tell you all, but it's slowly drifting away from us, very slowly. But that means that 4.5 billion years ago, that moon would have been closer. And as a result, our tides would have been much more powerful. They would have been pumping through our oceans, driven by a closer moon. There would have been lots of hydrothermal activity, we think. So hydrothermal vents would have been everywhere, lining the bottoms of our oceans. There would have been no oxygen, because that oxygen that we are breathing right now in our atmosphere was largely created by microbes capable of oxygenic photosynthesis. And asteroid strikes would likely have been more frequent. So overall, the surface of our planet, not really a friendly place to be. So the question becomes, where did life begin? What was the right, perfect place for that crucible of the first steps for molecules from rocks to slowly start making copies of each other, which slowly became the first cells and the ancestors of all life on Earth? How did that happen? Spoiler alert, we don't really know. And second spoiler alert, we kind of think the whole planet might have been involved. This is from a paper that several of us from the program here wrote, arguing that the whole planet might have been involved. There's no one single perfect place for life to have arisen. Those tides would have helped mix things up on the early Earth. And that's something to think about when trying to consider whether life could exist elsewhere. What's the exact right set of conditions and ingredients you need to begin life on a given body? But because of who I am, I want to focus on one particular place in particular, and that's hydrothermal vents, because we do think they may have played a special role in those earliest steps to the origin of life on Earth. And I have to give a shout out to John Barrows, who was one of the very first people to suggest that this may have been where life originated on the early Earth. When he first suggested this, everyone thought that was crazy, but now it's one of the more accepted theories for where some of the most important steps for the origin of life may have happened. To give you an example of why they're so important, well, one, just to go back to these vents, they have, the most important thing I want to tell you is that in the middle of these vents, that water can be 400 degrees Celsius, so 4 times the boiling point of water, with an extremely low or extremely high pH, and chock full of all kinds of chemical compounds like iron and sulfur and hydrogen, even things like methane and formate. But then if you move just a few feet away from that hydrothermal plume, that water is 2 degrees Celsius, and it's an entirely different chemical composition. And then as you move closer, you have everything in between. So we have all kinds of environmental gradients. That means you have all kinds of, it's like a natural laboratory, all kinds of different conditions that you can use to play with for those earliest steps for an origin of life. Some have postulated that the interior structures of these hydrothermal systems could have all kinds of perfect pores and nooks and crannies to get some of those natural experiments happening. Some people have even cut away hydrothermal vents and done modeling to see if you could concentrate nucleic acids in just the right amounts to get things together to form sort of a prebiotic soup. And some have even argued that these little fissures and pores could have been the perfect place to have the first cells, that the first cells were not in fact encapsulated by lipid membranes, but instead were inside essentially rock cells, and that life essentially had to escape from these hydrothermal nurseries to become the life we see today. That's one theory. The big question here that we're trying to ask is, how did life begin? And the actual answer is we have no idea. But we are trying to figure out ways that we can try to approach that question in a scientific manner. And we can do that through two broad approaches. The first is bottom-up approaches. So basically, working in the lab, trying to recreate what those earliest conditions may have been like, making experiments, trying to put together replicating molecules to see if they'll make copies of each other. Those are called bottom-up experiments. But we can also work in the other direction. People like me are looking at modern life as it exists today, trying to look at what we all have in common, and then working backwards to try to think about whether we can infer something about what our last universal common ancestor, what those earliest origins may have looked like. So I'm going to focus a little bit more on those top-down approaches. We just ask ourselves, what does all life have in common? What can we learn from that? One of the things we all have in common is what we call the central dogma, or the unity of biochemistry, which is that, so here's a little intro bio for you guys. We all have DNA. That DNA gets transcribed into RNA. That RNA gets translated into protein. And proteins are the workhorses of our cells. All life on Earth has that in common. There's that, and there's these things called ribosomes. And I think I got a little too big for my britches here, and I tried to make a fancy video that I think is going to fail horribly. Ooh. Mute it first. Okay. So you guys get to watch me flail around with technology here. All right. So here's an example of a ribosome. Ribosomes are molecular machines that we all have inside our cells. Those molecular machines are what are turning RNA, or translating RNA, into protein. Those molecular machines, one of the most fascinating things about them is they're kind of like molecular fossils. We can look at them and try to infer something about what the earliest molecular machines may have looked like. And one thing all of these molecular machines have in common is that it's RNA that's actually powering, catalyzing these reactions that are common to every single living thing on Earth. And we can learn from that. So RNA, to remind you all, so here's DNA. Here's RNA. RNA is special because not only can it encode information, which is important for passing on information to the next generation, but it can also fold up into special shapes. And in folding up into special shapes, it can push chemical reactions forward. It can act as a catalyst. So it has this dual nature, which has led people to think that possibly RNA may have had a special role to play in those earliest stages of the evolution of life. That it may in fact have been RNA that was folding up into what we call ribozymes and making copies of other RNA molecules, and that those earliest tiny little molecules may have been the precursors to all of life as we know it today. So just to give you an example of what that may have looked like, you could have had these ribosome type molecules making copies of each other. Some of those copies were imperfect or different from the copies that came before. Those slightly different copies may have been better at making copies of themselves, and so on and so forth. And suddenly we have evolution going on. That's all evolution is. It's things that are making copies of each other. Some of those copies turn out to be slightly different, and some of those slightly different things are better at making more copies and surviving and reproducing and so on. So we're going from, these tiny little molecules all the way out to the life as we see it today. One of the first things that would have happened is we would have had the last universal common ancestor to all of life on Earth emerge. We call that Luca. And it would essentially be your great-great-great-great-great-great-great-great-great-grandparent. And a lot of scientists have been trying to work out what Luca looked like. What did that last common ancestor that we have for all life on Earth, what did it actually look like? There have been some really interesting studies lately that have examined all life as we understand it on Earth, try to look at what we have in common and infer something about what that last common ancestor may have looked like. Some think that it may have lived in hydrothermal vents, that it has just the right combination of molecules inside it that would adapt really well to a hydrothermal vent ecosystem. Not everyone agrees. There was a paper that came out last year that suggested that that last universal common ancestor to all life on Earth was actually fairly complex. It had a pretty big genome. And what I found most interesting was that it probably had an immune system. So just as we're all still dealing with viruses, flu, RSV, COVID, our earliest ancestor had to deal with it too. So we've been dealing with this for millennia, which I find fascinating, and that it may have actually had a fairly complex immune system. So what I find so fascinating is that this is something that happened billions of years ago, but just by looking at life as we understand it on Earth today, we can try to infer something about what those earliest ancestors may have looked like and where they may have lived on our planet. That last universal common ancestor to all life on Earth would eventually have diversified over time to become the life that we see today. This is a somewhat modern view of the tree of life. So that last universal common ancestor would have been right there in the middle of the tree. We are somewhere out here on the edge of this tree now. And as that life diversified and evolved and changed over time, it would have changed what our planet looked like. Even the tiniest microbes would have had a huge impact on the whole planet. Let me give you an example. Some of those earliest microbes, we think, were probably called what are called methanogens, meaning they produced methane. Today, we see methanogens in things like the guts of cows or in marshes. We can think methanogens for the fact that cows are producing farts that will contribute to our greenhouse gas problems. Methane is a very powerful greenhouse gas. But in the earliest days of Earth's evolution, some of those earliest microbes may have been producing methane. That methane could have escaped into our atmosphere and helped contribute to greenhouse warming on the early Earth. And in fact, some here have suggested that that early Earth may have had so much methane that may have created something like a methane haze on that early Earth. And so if you were an alien looking at Earth through a telescope billions of years ago, you may not have seen a pale blue dot, you may have seen a pale orange dot, and it may have been in part because of the microbes that were producing methane on that early Earth. So here's one early example of how those tiny microbes could have had a planetary scale impact on what our planet even looked like. And that continued through Earth history. So, oh God, why do I keep doing this to myself? I keep getting all fancy and making videos. Mute that, push play. Oh, I don't have a play button. It's in that one. I know how to use YouTube, I swear. What I'm trying to illustrate are cyanobacteria. So this would have been one of those evolutionary innovations that happened about 3-ish billion years ago when some clever microbes came up with the ability to harness the power of water, to crack water open using the power of the sun and create oxygen. And that oxygen would have done things like rust out all of the iron that would have been in our oceans. That rusted iron could have sunk to the ocean's depths and created what we call banded iron formations, these gorgeous rusty formations we see in various places on Earth. It would also have, eventually, once the iron had rusted out, that oxygen would start to accumulate in our atmosphere. And so our atmosphere would slowly have become oxygenated. This is a figure showing time, so age. This is the present day. This is 4 billion years ago. This shows how much oxygen there was on the Earth. And you can see there would have been this massive jump a little while after life invented this new ability to metabolize water and turn it into oxygen and create energy in the process. So here's an example of life fundamentally changing our planet because of these new evolutionary innovations that were appearing. Oxygen is an extremely reactive gas that would have changed our planet in fundamental ways. First of all, any of the life that was currently living on our planet at the time was not adapted to oxygen, so probably would have died. So it probably would have led to a mass extinction event. And on top of that, some have theorized that oxygen could have scrubbed the methane out of the atmosphere, removing a really powerful greenhouse gas and plunging our planet into a snowball Earth, where the entire planet was a giant snowball. This actually, we think, happened multiple times in our planetary history. I know there's still a lot of debate about what caused it, how we got out of it. Ask people in this room. I'm not an expert. I just think it's fascinating that something as small as these microbes could have contributed to the entire planet icing over. I mean, that's just incredible to me. Eventually, and here comes another YouTube video. Why do I do this to myself? It's because I love David Attenborough. That's why. Okay, hang on. I can do this. No, it can't. We're skipping it. Okay. What I was trying to show you, and I'll just show you this picture, was that the release of that oxygen that was first initiated by those microbes long ago that invented the ability to do photosynthesis paved the way for the evolution of complex life on Earth. We would not exist. We would not be able to form these multicellular creatures that we all know and love, again, the charismatic megafauna, without the invention of oxygenic photosynthesis that, again, paved the way for life as we know it today. Just imagine otters and penguins playing around. Okay. That life continues to impact our planet today. So here's an example of what our biosphere looks like now. This simulation shows what the Earth looks like over the course of a year. It turns out that there is more life in the northern hemisphere of our planet than there is in the southern hemisphere of our planet. All the plants in the northern hemisphere of our planet, of course, are going to be growing like bonkers. Trust me, if you're from Minnesota, you would know the whole thing turns into a jungle in the summer. And in the winter, it all dies. And so we have oxygen being produced like crazy in the summer from all those plants. And then in the winter, and then as you get into the fall and spring, those the dead plant matter is decomposing and releasing carbon dioxide. So over the course of a year, you have these cycles of carbon dioxide and oxygen happening. And here I'm showing you carbon dioxide over the course of a year. And what you can see, if you're patient, is that the carbon dioxide will become more intense, more concentrated in our atmosphere as you get into the autumn. And then it becomes drawn down in the summers when all the plant life comes back. So basically, our Earth is breathing over the course of a year as we go through the seasons. These are the kinds of things that happen to our atmosphere when you have this much life in our biosphere that's producing these gases and consuming these gases that have effects on planetary wide scales. This, of course, is interesting to astrobiologists because these are the kinds of things that we can see with telescopes that are indications that there might be life on a planet. I mean, yes, we know there's life on this planet, at least I think there is, but we can use that information to look for life elsewhere. So I stole this from Jada. So I'm moving into the deep space part. So here's where I pretend to be an astronomer. So just, grain of salt. But Jada gave me this image and she likes to argue that our Earth is more than one planet because it has changed so much over the past 4.5 billion years. And we have to keep that in mind when we're thinking about how to look at other planets to see if there's life on them. They're not going to be the same as they evolve through their own planetary histories. So if we, again, were to imagine an alien with a telescope looking at our planet, it would look very different depending on whether it was looking at it today, 2 billion years ago, or 4 billion years ago. And the same applies for us looking at other planets and trying to figure out whether there might be life on those planets. How do we figure out whether there might be life on those planets using only a telescope? Well, we can look at the spectra of those planets. We can take the light coming off of those planets from their atmosphere and break it up and use those spectra to try to figure out what kinds of compounds, what kinds of molecules might have been present in the atmospheres of those planets. So to break this up a little bit more, if we go back to that pale blue dot image I showed you before, if we were to or use a telescope to look at that pale blue dot, break up that light by wavelength and look at the dips and valleys that occur in these spectra, each one of those peaks and those valleys corresponds to a different molecule that exists in that atmosphere. And we can use that information to try to infer something about what might be present on that planet. And some of those peaks and valleys might correspond to things like, for example, methane that we think largely, not always, but largely can be produced by life. We call those biosignatures. Now, some of them can fool us. Things like oxygen, we may think might only be produced by life, but sometimes it can be produced by abiotic things as well. And so that's why people like here at the University of Washington are trying to figure out, okay, which of these gases is produced by life and in what context and when, and how does that change over time? And how does the environment change, whether that biosignature is going to stay in the environment or whether it's going to be, consumed very quickly. Those are the kinds of questions we're trying to ask ourselves. Both in the present day, trying to understand what the Earth would look like in the present day if you were to take that spectrum and break it up into its bits, but also what it might have looked like 2 billion years ago or 4 billion years ago. And here's where I really try to pretend to be an astronomer. So one of the things that NASA is working towards now is to actually look at other exoplanets beyond our own solar system and really for the first time start to look at those atmospheres and figure out whether we can find signs of life on those planets. So what I'm showing you here is the Habitable World Observatory is NASA's one of its next flagship missions. It was recommended by a decadal survey a few years ago. It's essentially going to be a telescope that is going to be the first to actually image these exoplanets and look at their spectra, so we can try to figure out what kinds of gases, what kinds of molecules are in those atmospheres. And as a result, is one of the first telescopes that's really been designed to search for signs of life on planets outside of our own solar system. If you have questions about HWO, you should ask Vicky, who is here and offered to answer questions about it. But what's exciting is that we're bringing together the knowledge of people who are oceanographers and biologists and chemists and atmospheric scientists and astronomers to ask these really big picture interdisciplinary questions and apply them to these flagship missions to really, really in earnest continue that search for life beyond our own planet. I'm going to show this slide as a way to kind of, end here. I'm only 35 minutes in. That's okay. Lots of time for questions. And say that I feel really privileged to have been one of the graduates of the astrobiology program here, because it really trained us to think in a really big picture way. I have learned how to feel really stupid every single day, but to feel okay about that. Because I was trained as a biologist, I was trained as an oceanographer, and when I'm talking to astronomers, we have to learn how to speak the same language so that we can work together to ask these really big picture questions. And so it's totally fine for me to say, I have no idea what an M dwarf star is. Can you please explain that to me? And by learning how to speak the same language, we can move forward together to ask these scientific questions. And many of my closest friends and mentors and colleagues have come from this program. Most of the people you see here from these really ancient photographs are still my close friends, mentors, and colleagues. And I feel really privileged to have been able to work with them to ask these really big picture questions about life in our universe. So thank you for your time, and I'm happy to take all this time we have left for questions. Thank you.
How do we ask a question?
Questions? I will run the mic to you.
I'm watching from home. I'm not there.
Okay, I'm coming. You can run the other mic if you want.
Thank you, Rika, for a very wonderful talk. This is Gio over here. I was wondering, you mentioned that Luca is thought to have had a immune system. Is it known whether that immune system was against RNA or DNA viruses?
It's a CRISPR system. And so that could, I think that is independent of whether it's an RNA or DNA virus. We don't really know whether RNA or DNA viruses came first. I will admit I was a little skeptical when I saw that result from that paper last year. I think CRISPR, to explain to the rest of you, you've probably heard of CRISPR-Cas9 as something that genetic engineers are using to engineer new types of life. But actually, what it really is, where it originally came from, is it's an immune system in microbes to recognize viruses that have infected those microbes in the past. I love using them as sort of a library of previous viral infection that I can use to study which viruses have infected which microbes in the past. And a study was done last year to try to infer what our last universal common ancestor may have looked like. And in their paper, they argued that that last universal common ancestor would have had this pretty sophisticated immune system that we call CRISPR. I am a little skeptical. I wonder if it may have been that their methods, because CRISPR is so widespread, it's found in so many different types of microbes because it's so useful. I wonder if, to use a technical term, horizontal gene transfer may have muddied their analysis a little bit. And I wonder if it actually isn't as ancient as they claimed, but I don't know. So to answer your question, probably both, but to be determined, I think.
Next. Okay, I have a couple, but I want to start off with a kind of silly one, right? So, okay, is this better? Okay, so I have a couple of questions, but I want to start off with a kind of silly one. Do you have a collection of tiny styrofoam cups? Because I've heard that this is the thing that like oceanographers and people who send probes to the deep ocean do. So they'll attach styrofoam cups to like the probes and stuff because the pressure will just compact them. And then people will like keep collections.
I do have a collection of tiny styrofoam cups. Any self-respecting oceanographer has a collection of tiny styrofoam cups.
I've been told the number of cups is how you determine hierarchy among oceanographers. Is that true?
No, it's who designed the coolest styrofoam cups, I think, is what determines your hierarchy. So for the rest of you, so what we do is we put styrofoam cups into these mesh bags. We stick them to our instruments. This is a very important part of the daily scientific process. We send them either on like Alvin, the submersible, or on a CTD package that we use a wire to send to the bottom of the ocean. The immense pressures at the bottom of the ocean will squish the air out of those styrofoam cups and make them like, a cup that was this big will shrink to about this big. The cheaper the styrofoam cup, the better, because they have more air pockets to squish. The things you learn, right? Was that your question?
Yeah, that was my main question.
Okay.
And my other one was like, what do you think of Titan? Saturn's moon?
Oh boy. Okay. Titan is one of the moons of Saturn.
Yeah, Saturn.
And it has lakes and oceans of hydrocarbons. And some people really, really like Titan because it's full of organic molecules. And when you think organic molecules, you think life. And so some people think that Titan could be teeming with life because it's got these like liquid oceans that are just made of the ingredients that make life as we know it today. However, those lakes and rivers and oceans are probably like 80 degrees Kelvin, super, super cold. And so I don't know, I'm not a Titan scientist, so I hope nobody in here is either. I apologize if you are. But I apologize if you are.
Someone, so.
I just sometimes think that those are really low temperatures and it might make it hard to push chemical reactions forward. I'd want to know what catalysts were there. It's a fascinating body, that's for sure.
Mm-hmm. Come on.
Awesome. Thank you for your talk. And I was wondering, the prevalence of oxygen in an exoplanet's atmosphere, how likely is that to be an indicator of life, or could it also just be from some other process? Like if we do find an abundance. Yeah.
She's the world expert on this, so I'm going to cede the floor to her.
Yes, I once wrote an entire, I don't know how many page paper on oxygen. Okay, so how likely is it to be? Well, it will depend on what else is going on in the system, what other things you can learn. So what my research team, the Virtual Planetary Laboratory, did is we actually learned that there were multiple ways you could make oxygen without life. And as I explained in last week's lecture, if you're there, cyanobacteria and plants on our planets will produce oxygen. And so for a very long time, it was considered that abundant oxygen in a planetary atmosphere would be an indication of this photosynthetic biosphere that we have here. However, things like Rika explained that, our sun at least was fainter in the past, but other people's suns around M dwarfs, for example, are brighter in the past. They can boil off oceans. And when you do that, you end up with a massive oxygen atmosphere. So one giveaway then would be if the oxygen atmosphere is due to an ocean loss, for example, we would see lots of high pressure oxygen bands in there. So that would give that away. In other cases, if we have the breakup of water vapor or CO2 in the atmosphere, that can sometimes create oxygen without life being there. But then we see the other bits and pieces of those reactions hanging around. So the general answer to it is yes, we could generate oxygen from other types of processes, but as we learn about these processes, we look for the tells. We look for the other things that are happening in the environment that tell us that oxygen is due to this geological or photochemical process rather than life. And so the trick now is it's not just about detecting life, it's about detecting all the other things that would tell us whether or not that oxygen is due to life or not. And so because of that, HWO, when it is built, we will be thinking very carefully about what other molecules can it detect, not just oxygen, but the other things in the atmosphere, like carbon monoxide, for example, or abundant CO2 that might tell us the oxygen is due to the breakup of carbon dioxide rather than being due to life. So I don't know the probability that when you see oxygen, it's not due to life, but we certainly have the techniques now to try and determine if that is true.
Hi, thanks for a great talk. You mentioned a bit about biosignatures, and a lot of those biosignatures were kind of based around assumptions of Earth life. And I'm glad that someone mentioned Titan a bit earlier, because it's a great example, but it doesn't have to be Titan. I'm curious how you start to I guess, train yourselves to be creative, to look for biosignatures that are completely different from anything we might find on Earth?
That's a really good question. And I think there's a lot of ways one could answer that question. I'm sure an astronomer would give a different question to what I would say. The first thing that I have to remind myself of is that our best example is the life we know here on Earth. And so we know for sure what gases are produced by microbes here on Earth. We know for sure how those, well, we don't know for sure. We're trying to figure out how the gases produced by life on Earth would have changed over time. We have an N=1. I mean, that's the truth of the matter. At the same time, we do try to have conversations. We had many conversations as graduate students in this program about alternative forms of life. John wrote a whole book about weird life, alternative forms of life, what kinds of alternative chemistries there could have been if life didn't use the unity of biochemistry that I talked about earlier, or could there be other metabolisms that we don't know about producing other gases? A lot of that is, I mean, some of it's speculation, a lot of it's grounded in good science. And so we do try to keep that in mind. But when we're designing these kinds of missions, and again, astronomers can speak more to this, we do have to rely somewhat on the life that we can study and really understand. And so we're both trying to work from what we know and be creative within those parameters. I don't know if that's a satisfactory explanation, but we do think about it for what it's worth.
Good enough.
I think we have questions on Zoom.
Okay, Ivy has a question. Hi, Rika. Thank you for the insightful talk. My question is on microbes. How different are these microbes in and near the hydrothermal vents versus the ones on the deep ocean surface?
Maybe she means, or this person means on the ocean surface. Okay. So we see different types of microbes. So the species that we see living near the hydrothermal vents are different from the ones we see living at the ocean surface. I think maybe the most interesting thing for folks asking that kind of question is what are those microbes eating? What are those microbes breathing? It's going to look very different at the surface of the ocean compared to in these hydrothermal vents. At the surface of the ocean, we have light, we have lots of oxygen, we have colder temperatures relative to the hydrothermal vents where we have much less oxygen. We have all kinds of compounds like hydrogen and sulfur and iron and manganese and carbon dioxide and methane that aren't as abundant at the surface of the ocean. So what the microbes are eating and breathing is very different depending on where they live. And of course, there is no light. I mean, we always, when I show those videos of hydrothermal vents, they're these gorgeous things. We have to remember that's only what they look like when they're bright, shining lights blinding all of the things that live in those hydrothermal vents, right? Usually they're in complete darkness. So they don't have the benefit of light to power those ecosystems. There's all kinds of other differences. They have to find ways to tolerate the high temperatures in these systems. The viruses even look different. That's one of the things that I study. My favorite thing is that, so I study viruses that live in the oceans. Most people think of viruses as something that would only infect humans. I mean, we all know COVID all too well by now. But actually, if you took a teaspoonful of seawater, there would be 10 million viruses in that single teaspoonful. And most of them wouldn't be infecting humans. They would infect the microbes that live in the oceans. If you took all the viruses in our world's oceans and lined them up end to end, they'd reach across the Milky Way galaxy 100 times. It's a lot of viruses. What are they doing? That's what we're trying to figure out. And it turns out in hydrothermal vents, it seems like they have a slightly different lifestyle than the viruses that live at the surface of the ocean. I should say, there's more of a certain type of virus that lives in these vents. There's a kind of virus that can like worm its way into your genome. So it takes its own DNA and puts it into the microbe's DNA, and then it can just hang out for several generations. And while it's doing that, it can mess with the microbe. It can mess with its host by producing proteins. It can change the way it behaves or change what it can eat, what kind of energy it can access. There are more of those types of sneaky viruses in hydrothermal vents than there are at the surface of the ocean. So how is that messing with the microbes? How is that changing their evolution? How is that changing their ecology? That's one thing we're trying to figure out. So the answer is lots of things are different, but lots of things are the same too.
Hi, thank you for this. And I want to ask, is there any other ways like expect we learn the components in our own ocean and we sending space telescopes into the space to learn the spectrums from other stars? Is there any other ways for us to learn astrobiology stuff?
And so the question was, are there other ways for us to study life beyond Earth besides looking at spectra through telescopes? Was that?
And also from looking from our own planets.
Yeah, I mean, so one of the ways we search for life elsewhere, especially distant exoplanets, is using spectra to look for biosignatures, like I mentioned before. And that's partly just because of necessity. They're really far away. It would take a long time to send something to those exoplanets beyond our solar system. Within our own solar system, it's closer to home. And so we can actually send robots, rovers, to those other bodies to try to actually take samples or go into the atmospheres of those bodies. I know that actually somebody mentioned Titan. I think there's a mission that's being sent there to send like a quad or octocopter or something. I forgot. Oh yeah, yeah. Well, yeah, it's called Dragonfly, but it's like this drone with all kinds of rotors that they're actually going to send to Titan to look at those lakes and rivers and oceans on Titan. So we can, within our own solar system, send missions to Mars or to Titan or to Europa or Enceladus to actually, get a closer look at those plumes and get a closer look at those oceans. I'm not sure if I'm answering your question, but there are different ways we can look for life beyond Earth. Did that answer your question? Is that what you were asking? Sort of. Sorry, I wasn't sure if I was interpreting that right.
It's like, because I feel most of the time we're learning astronomy, it's like we're using telescopes. And I feel like, is there any other ways because you combine biology into it?
So the way, maybe what you're asking is how biologists can contribute to the study of astrobiology.
Maybe it's like I just want to know, is there like any other aspects like you can learn the subjects?
Well, okay. So I think what I'll say is that to do astrobiology, you need to bring together all kinds of different disciplines. And so like I'm a biologist, I'm studying the bottom of the ocean so that I can try to get a better understanding of how life began and where life began. And then we can use that knowledge to think about whether life could begin on a specific type of body or moon beyond our solar system. So I guess there's a lot of different ways to integrate different backgrounds to the study of astrobiology. I'm not sure if I'm answering your question, but we can also chat afterwards if you'd like to.
Thank you for a great talk. When you presented the time series of gases in the atmosphere, first of all, there was a long period where oxygen levels were relatively constant, and then they went up about 500 million years ago, I think. And also the ozone, there was essentially no ozone until that time, and then it went up. So there are two questions. Why was it steady for so long and then rose? And what did the implication of no ozone have for the upper 10 feet of the ocean and life on land?
You know what? Are there any geologists here? I can't remember why that second oxygen rise happened. Anybody, any geologists here who can tell me? Because I actually don't remember. I'm so focused on the great oxidation event that I've forgotten that second rise. Are you answering the question or asking one? Okay. No geologist here? Wait. Charismatic megafauna again? That's why I didn't know. So I'd have to check with a geologist to answer that, or apparently a forestry person to answer that question. For ozone, I mean, that is something we talk about in the context of early life, that we wouldn't have had oxygen in the atmosphere and therefore no ozone in our atmosphere. And so when we think about various scenarios for settings for the origin of life, we often discuss what impact ozone would have had or the lack of ozone would have had, and therefore the strength of UV radiation, and whether that could have destroyed prebiotic molecules on the surface of the earth, which is part of the reason often people tend to favor like a little bit deeper in the ocean, because it would have provided some protection from that extreme UV radiation. So it's actually a really good question. Yeah. Yeah. Oh, please.
I'm just going to add to that because that's something Jada studied, and she's not here, and so I feel like I'm gonna channel Jada here. But in this very top plot here in the Archean Earth, can you see there's a label that says haze? So even though we didn't have ozone, and in fact, because we didn't have ozone back in the early some geological evidence and also Jada ran photochemical models that showed that you could in fact form basically a hydrocarbon haze early on. And that big dip in the orange curve there is that hydrocarbon haze absorbing ultraviolet radiation in a similar way that, not exactly the same, but similar, the way that ozone would do that. And so in that early Archean time, Jada showed that we would have potentially gotten significant UV protection at the surface from the haze itself if the haze was there. So there's more than one way to protect the surface of the planet. But to emphasize Rika's point as well, you can escape, massive solar flares if you just get a few meters down into the ocean, and so that can be a huge protectant. Just ocean water is a huge protectant for ultraviolet. So life on land would have been hard potentially, but maybe the haze would have protected it. But certainly in the ocean, that was definitely a refuge area for anything wanting to escape ultraviolet or stellar activity.
Hey, Rika, thank you so much for this talk. I'm in Bob Morris's lab here in the astrobiology program. I'm here, sorry. And also studying deep sea hydrothermal vents, host virus interactions. So I really love this talk. A lot of the time, I feel like in the astrobiology community, the top-down approaches for phylogenomics have a caveat of, let's say, evolutionary choke points or mass extinction events that may limit our resolution back to Luca or maybe pre-Luca all the way back to origins. I was wondering what your perspectives are in terms of top-down approaches and their relevance to some of those really early time points given some of these constraints.
Yeah, I mean, one of the things I was going to say when I was talking about Luca and showing that tree of life is what you're seeing other survivors. There were tons and tons of dead ends, species that emerged that just didn't make it or were pushed off by a mass extinction event. And all we can see by looking at life as it exists today are the survivors. So we're missing a ton by using these top-down approaches. I use DNA sequences to try to infer something about what life may have looked like. So I'm very much dependent on life as we know it today. And we know we're missing a lot, but that's just a known limitation of the method that we have to be honest with ourselves about. Paleontologists can look at the fossil record and see evidence of now extinct forms and infer something from that. But the DNA record, we really are limited to what we can see now. And I think one of the images I showed before was in sort of implying that using those top-down approaches, we can really only go as far back as Luca and not really beyond that, because I imagine before that last universal common ancestor of all life on Earth, there would have been tons of evolutionary experiments happening, most of which led to dead ends. Luca, that last universal common ancestor, was just the one that made it. So we are really descended from the survivors throughout the entire history of life on Earth. And when we're using those top-down approaches, we have to be honest about our limitations, but that's something we have to do all the time, right?
Thank you.
Hi, my name is Marcus. Thank you so much. This was lovely. My question is actually related to the previous question. And I guess what I wonder, I know there's no way to know these things, so I guess I wonder if there may be any theories or thoughts or views of it. But what I wonder is, do we suspect that life formed in one location and spread everywhere, or maybe occurred in many locations? And then a sort of subset of that might also be, if we look at RNA and DNA, obviously, like you're saying, we're the ones who survived, and so we might say, well, clearly RNA and DNA is, given our planet, this is the right way that life would form. But do we have any, I don't know, ideas like, could it have been that different forms of life could have happened, but over the course of 4 billion years, it just so happens that DNA and RNA-based life was the one that took shape and took the lead, and everyone else sort of fell behind?
Okay, so that was sort of two excellent, very big questions. The first question is whether life began in one place and then spread, or whether it could have begun in many places. And I would counter with saying, how do we define life? That's the first thing I make my students do in a class I'm teaching right now, which I taught this morning from my hotel room, on the origin and early evolution of life. The first thing I have them do, and I'll answer your question. The first thing I have them do is read a paper by my colleague Lucas Mix, who is a evolutionary biologist and ordained Episcopalian priest who came from University of Washington, where he tries to define what life is. And I make my students try to define life, and they get all tied up and twisted around, and nobody can agree. And usually by the end of it, they're like, there is no definition of life. There's no point. And the reason I say, how do you define life, is if in one part of the earth we had RNA molecules forming, and another part of the earth we were synthesizing peptides, and another part of the earth is where we started to get those things concentrated together into a little pocket where reactions started to happen. what do we call the origin of life, right? My personal bias is that we did have many different locations on Earth that were part of that process. At what point you call it life emerging is sort of an open question. I do think, I mean, I have no way of knowing because I wasn't there. But I do think it's likely that there were lots of biochemical experiments happening in those early stages. Some of those biochemical experiments failed and some of them succeeded, and we are the results of the ones that succeeded. Some have postulated that some of those early biochemical experiments carried on in parallel evolution to ours to form what's called a shadow biosphere. We just don't know how to look for it. It's kind of an untestable hypothesis if we don't know how to look for it. But I do think that there were probably lots of different experiments happening, and we were the ones that made it.
Did you have another? We have another question on Zoom.
Okay, from Maliko. Are there other chemical, temperature, pressure, solvent systems that hold promise for complexity that could yield life?
Are there other, okay, so I'm,
even though that's what you study, I think she's asking, is there something besides hydrothermal vents that are complex that could
have yielded like I mean, I know you didn't ask the question, but could have yielded like an origin? Oh, yeah, absolutely. So this morning in the class I taught over Zoom from my hotel room, I made my students have a discussion about different settings for the origin of life. And of course, I assigned hydrothermal vents as one of those settings, but I also assigned them sea ice, and I also assigned them beaches, and I also assigned them warm little ponds, as Darwin put it, to try to look at the pros and cons of each one of those locations. And my class didn't agree on what location they liked the best for an origin of life. I like sea ice, and here's where I shout out Jody. So we have researchers here at the University of Washington who are studying life in ice. My favorite thing that Jody told me was that as seawater starts to freeze, all of those salts, but also all of those microbes and those viruses and the DNA that's in the water starts to get pushed into these little brine channels within the ice and forms what she called an evolutionary playground to get all these things together to exchange DNA and interact with each other in really interesting ways. And we could imagine that something similar may have happened on the early Earth if ice existed, which is another question, that could have pushed together the right prebiotic chemical compounds into these little sort of like almost like protocells potentially to make certain chemical reactions happen. Some people like sea ice as an example for where you could have pushed forward some of those early prebiotic reactions. Others have said very similar things about terrestrial hot springs, kind of like Yellowstone today, would have other conditions, wet and dry cycles that help polymerization happen. Those could have been really important for those steps in the origin of life. So I think to answer the question, there are a lot of different things that different settings can offer that could have contributed to forming the earliest cells and the earliest life is leading to what we know as life today.
So I guess I'm curious about, and I'm asking you to elaborate on the cycle of RNA creating copies of each other, the mechanisms of that, and how that leads to the formation of the first like cells and cellular structures, as well as how you're theorizing about them having to escape from these hydrothermal vents and how that would happen in the absence of a lipid layer.
The short answer is we don't know. I suspect that's not a satisfactory answer. In its purest form, what I roughly described as what is sometimes called the RNA world, which is this idea that the earliest replicating or self-replicating molecules or entities would have been RNA molecules that could both encode information and also make copies of themselves because of the way they can fold and catalyze reactions. That idea has changed and morphed over the years, and people now are more in favor of sort of an RNA peptide or RNA protein world where RNA and proteins were working together to make sort of molecule A will copy molecule B with the help of molecule C, which will then copy molecule D, right? And so all these molecules are working together to make copies of each other. At some point, those molecules that are working together would have had to sort of stay together, whether it's on a mineral surface or inside, whether, a hydrothermal vent pore or something in sea ice or even an early lipid bilayer. Lipids can actually quite easily form little like micelles and lipid bilayers in solution. I don't think it would have been that hard to form little bubbles that would have had molecules inside them making copies of each other. The question is how all that came together and where all that came together. There's a lot of debate. You should see how heated debates get at these conferences. Everyone has their own favorite opinion and they think they are right. I'll just say, I don't know. We're trying to figure it out.
Question. So you're talking about life, but how do you separate? Say we want to find intelligence, right?
Okay.
So we've got life, we've got intelligence. Are there any potential gas markers that could identify the potential of intelligence, as opposed to knowing there's like a probability problem rather than an accident?
So Woody Sullivan is the person to ask, but I will do my best to answer that question. And if Woody wants to chime in, please feel free. My first question is define intelligence. And do we have it on this planet? I don't know. I think when we as astrobiologists talk about this, we're not looking so much for markers of intelligence because again, what is intelligence? But we can look for markers of technology. And so we look for what are called technosignatures. And there are various things one can look for, like, evidence of radio waves and radio communication that one can look for to see whether life on a given planet has evolved to the point where we recognize forms of communication that we use ourselves. Is there anything you want to add, Woody?
Yes, well, that's a very good answer. I would just add, we can look at the evidence in the same spectra that things like the Habitable Worlds Observatory, we hope will get funded and eventually actually take a look at planets. We can look for spectra of chemicals that we can think of no natural way they could have been formed, except they're in industry. Chlorofluorocarbons is a good example that we've had trouble with on Earth. And there could be many others, unknown spectral lines. It would take a long time to figure out what it is. And you finally conclude that it can only be made artificially by something that was intelligent. That would be a long argument.
Well, just talking. I learned about Luca for the first time tonight. In the history of life, where do we think Luca falls? Kind of early or late?
Oh, quite early. That paper that came out last year that I was interviewed about like 20 times, so I'm very used to talking about it, they estimated that Luca emerged about 4.2 billion years ago. The Earth formed about 4.5 billion years ago, so that is immediate. There's some skepticism that it was that early, but even when you look at the rock record, there is evidence that life began fairly early. And that's one of the most remarkable things I learned here about the evolution of life on Earth, is as soon as the planet became habitable, life formed. Now, that's n equals 1, but it's an interesting conclusion when you're thinking about the possibility of life elsewhere.
Hi, hello, hello. Okay.
I can repeat it, I can hear you.
So,
two different planets, what way can you not contaminate them? For example, endospores, and how that affects our data.
Yeah. So you're thinking about planetary protection. And that is actually something that I know John has spent a lot of time thinking about. It's not my specialty, but what I can tell you is that a lot of work is spent thinking about how to sterilize these telescopes, these robots, these rovers before we send them to other bodies, because it would be the worst possible thing, both ethically and scientifically, if we were to contaminate, say, Mars or Europa before we even had a chance to study the life that might exist there independently of life here. And so I know, and I don't know the details because I do not work at NASA. I have not worked in planetary protection. As I said, I know people here have done that work, but they think very carefully about what techniques we can use to make sure we're working in clean rooms, what technique can we use to sterilize these instruments before we send them out, especially the ones that are going to, say, drill into Europa's ocean. I think part of the reason we haven't sent a probe to Europa to do that, aside from the expense and time and money, is the fact that we have to think so carefully about not sending our own life to Europa to then contaminate the oceans, and then, it would completely eliminate the signal. And then ethically speaking, that would be a problem, right? That's my best answer I can give for that, unless somebody who's in planetary protection wants to comment on that, but.
Hi. About that question, you mentioned that there are signs of life, like in the very early days of Earth's history, and I was wondering what those signs are. And you also mentioned non-oxygen life, so life that might have existed before plants, cells learn to metabolize water and, turn into oxygen. Were those signs of those lives, or are we just kind of speculating that they might have existed?
Yeah. So the earliest evidence for life on Earth comes in many forms. Probably the most common form is isotopes. So many of you may know that we have atoms that can come with different numbers of neutrons. And so even the same carbon atoms can weigh different masses because of the number of neutrons. Life is lazy and prefers to use the lighter versions of those atoms, those isotopes. And so we can look for evidence of life's laziness in our rock record. And we have found evidence of life's laziness through the isotope record extending back to, it depends on who you talk to, but extending back to almost 4 billion years ago. So there's the rock record. The paper that came out last year about Luca that dated it to about 4.2 billion years ago, it was based on molecular clockwork, which is what I do, among other things. We are trying to use